So we have now, I think Chelsea, you're next, right? Or is that the wrong way? Yes, Chelsea is coming next. She's doing biomarkers of cellular senescence and inflammation associated with exercise capacity among adult survivors of childhood cancer, and speaks to us from St. Jude's. So thank you.
Okay, so hello. It's lovely to be here. Thank you for the opportunity to share some of the work coming out of Memphis, Tennessee, where we're very blessed to work at St. Jude Children's Research Hospital, where no child is ever turned away based on their race, gender, religious belief, or socioeconomic status. So we're very fortunate to get to do some very exciting work there, and I'm a postdoc in the Human Performance Lab under Curie Ness, where we've recently looked at associations between cellular senescence and inflammation and exercise capacity among adult survivors of childhood cancer. So I think majority of people in this room understand what exercise capacity is and why it's so important. It's the ability of the human body to dictate function, incorporating central and peripheral organ systems. And as you can see here from the graph, we know that there's a plateau or a peak where our body maxes out based on what our systems can do. So it is possible for individuals to be exercise intolerant, so they're limited in their exercise capacity or their maximal ability to function from the cardiopulmonary system, and sometimes that can also be at the periphery, so delivery of oxygen to working muscles or even the muscles themselves to extract substrates and oxygen. And so here, these are just basic VO2 norms that we have in the general population, and you can see with males on the top and females on the bottom that with increasing age, we see a decline in exercise capacity. So what once was considered poor at age 18 is now considered average over 65 years of age. And so childhood cancer is important. We see that there's an increasing incidence over time. However, thanks to significant advances in treatment over the past few decades, 5-year survival rates can be north of 80–85%. So in the United States alone, there's over 400,000 childhood cancer survivors, and almost half of those are actually under the age of 40. And so a lot of the research that we do in the epidemiology and cancer control department is look at risk factors or late adverse outcomes in this population, and how might we intervene to improve childhood cancer survivors' quality of life. Unfortunately, we know that many of these survivors experience chronic morbid conditions later in their life, and this graph here shows that in the childhood cancer survivor study, so it's across the United States, in over 5,000 childhood cancer survivors at age 50, the average number of chronic conditions at a CTC AE grade of 3 or 5, so severe or disabling, is 4.7. And that's almost twofold higher than in the general population. So it's great that these kiddos are making it into adulthood, but now they're plagued by all these chronic conditions that are related to cardiotoxic treatments that they receive at a young age. And so we were interested to see whether exercise capacity was altered in this population. And so we investigated exercise intolerance, mortality, and organ system dysfunction in adult survivors of childhood cancer. So in this study, we were able to recruit from the St. Jude Life cohort study. So any child that's been treated at St. Jude since 1962 and is at least 5 years since diagnosis is invited back for routine organ system testing. That also includes physical function assessments in the Human Performance Lab. And so we were able to look at exercise capacity in over 1,000 survivors at a mean age of 35, and almost half of these, over half of these were exposed to anthracyclines and chest radiation, so those cardiotoxic therapies. We're also fortunate to be able to compare our results to healthy community controls recruited from families, friends of our St. Jude Life participants. And so we're able to compare our results to a normative population. And so we use the cardiopulmonary exercise testing and set our exercise intolerance value at 85% of predicted using the Friend equation. And so as you can see here, a large proportion of our survivors that were exposed to cardiotoxic therapies had exercise intolerance, and even survivors that weren't exposed to those therapies were also exercise intolerant, which was well higher than our community controls. And then we investigated the associations between exercise intolerance and risk on mortality. And so survivors that were exercise intolerant had an increased hazard ratio of 3.9. So this is very concerning for us, knowing that exercise intolerance impacts daily function in these survivors. And we also have knowledge of the impact of exercise capacity and cognitive reserve and daily functional living and longevity. So survivors that are exposed to cardiotoxic therapies are at increased risk, but there also seems to be something else, a pathobiological mechanism underlying this intolerance in survivors that weren't exposed to these cardiotoxic therapies. And so there is evidence in St. Jude Life and in childhood cancer survivors that there's this premature aging or accelerated aging phenotype that is occurring. And we know that cellular senescence is one of these possible mechanisms from the hallmarks of aging. This is a part of normal life with progressive age. It results as a loss of function at the cellular level, and the cells become unresponsive to growth stimuli. So they're not benign, they're just in a stable state of growth. And so as we increase with age, we see an increase in these senescent phenotype cells that have the ability to secrete high inflammatory cytokines, immune modulators, growth factors, proteins, etc., known as SASP, that can result in subclinical inflammation and contribute to body composition changes in this population. Specifically in white adipose tissue, we see an accumulation of these cells, and unfortunately many of our survivors, upwards of 40%, are overweight or obese, so this is a concern for us. Although these cells are largely undetectable in younger populations, we do see it in older individuals as well as childhood cancer survivors that were previously exposed to radiotherapy. So when they took skin biopsies, we saw an elevation of P16 mRNA expression. And so given that childhood cancer survivors are exposed to therapies that are capable of inducing DNA damage at the cellular level in normal healthy tissues and not cancerous tissues, it is possible that senescent cells might be underlying a phenotype that we see in exercise intolerance in our population. And so that was the purpose of this investigation, was to evaluate associations between cellular senescence and the subsequent inflammation and exercise capacity among childhood cancer survivors. And so there's a little shot there for you of our campus in Memphis, Tennessee, and our after completion of therapy clinic where the Human Performance Lab is housed. And so we recruited participants from the St. Jude Lifetime cohort study, and then for these analysis, our participants were adults, so they were older than 18 years of age and at least 5 years from primary diagnosis, and did not have any active cancer reoccurrence at the time. And so this is just a quick overview of the St. Jude Life cohort study. You're welcome to take pictures, obviously, because there's a lot of information, but as I mentioned, these survivors are recruited back to campus for a 3 or 4 day visit where they get all these medical tests, so we're able to keep an update on how they're doing functionally and in their organ systems. And then part of this assessment includes a human performance visit, so we were able to capture an exercise capacity from a CPET test in this population using a modified Bruce protocol. Survivors also get a DEXA scan, so we were able to capture body fat just at the 2D level. And then participants are also invited to give blood samples at the time of the visits, and so we were able to evaluate cellular senescence using P16 mRNA expression and C-reactive protein expression in peripheral T lymphocytes in this population. So an overview of our study population. So just about 56% were females and 77% were white non-Hispanic, and a mean age of 36 years of age. And primarily, we see acute lymphoblastic leukemia survivors and lymphoma survivors in St. Jude Life, given that they have the highest survival rate, and so they make up a huge contribution of this study. And just a brief overview of what their cancer and treatment history were. So we see that majority of the participants received a combination of chemotherapy and radiation, again, because we're across all the childhood cancer diagnoses, and over 70% received anthracyclines, which we showed earlier in the exercise intolerance study as a cardiotoxic therapy. And this here is the distribution of peak VO2 and body fat by gender, which is what we would have expected in our survivors and consistent with the general population. So we see compared to females, male survivors had a higher peak VO2, but a lower percent of body fat than females. Here you can see on the left-hand side the markers of inflammation, and on the right-hand side our P16 mRNA expression, which we log transform for analysis. So on average, females had a higher level of inflammation indicated by higher CRP concentrations, and more cellular senescence indicated by the higher P16 expression. And so we looked at multivariate models to evaluate the associations between these biomarkers and exercise capacity. So on the left, you see the regression model for males in red, and on the right-hand side, you see the regression model for females. And below are the results of the multivariate models. And so when we stratified by gender, you can see that we corrected for body fat percent and age at the assessment, so when they came to St. Jude Life for that functional assessment. And you can see here that inflammation was associated with declining exercise capacity only significantly in females. When we look at cellular senescence, so that P16 expression, again, with males on the left and females on the right, we can see that the slopes between the males and females were quite different. So when we ran multivariate models, again, accounting for age and body weight, we found a significant interaction between the P16 expression and the amount of body fat in females. And so to explore that a little bit further, we looked at varying levels of body fat among the females, and you can see here that the relationship between P16 and exercise capacity seems to level out as we get at those higher levels of body fatness. And so again, just reiterating here, as we increase the body fat percent, we see a more leveling out association between P16 and peak VO2 in our population. So in summary, these analyses show that exercise capacity, or suggest that exercise capacity is inversely associated with biological aging, so cellular senescence, as well as the subclinical inflammation in our population. And the relationship between cellular senescence and exercise capacity seems to be influenced by body composition, specifically body fatness in females. There is evidence, thankfully, in the basic science literature that suggests that it's possible to mediate or remove cellular senescent cells. The most notable example of this is in a mouse study by Marissa Schaefer and colleagues at Mayo Clinic, where they exposed mice to fast food diet, so that high-fat, high fructose diet, and then they exposed them to exercise, and they saw a reduction in these senescent cells with exercise on a treadmill running, as well as an improvement in the total amount of time that they were able to run on the treadmill. And so looking at our population, we don't know if they have the same capacity to mediate the accumulation of these cells, because we are still investigating whether the response to exercise is consistent with the general population. But these early impairments do suggest that we might be able to use VO2 as a biological marker of an aging phenotype in our population. So moving forward, we want to explore a little bit deeper the treatment associations, specifically identifying childhood cancer groups at highest risk, given that the treatments are quite different across the different populations, as well as I mentioned, are we able to remediate the accumulation of these cells in this population? So it's a huge team effort, epidemiology and cancer control, a lot of great mentors. We work closely with colleagues in cell molecular biology, biostatistics, pathology, and immunology, and our human performance lab is absolutely fantastic. We see about 40 functional assessments a week, including remote exercise interventions, so the team is absolutely fantastic, and we appreciate all the support from the National Cancer Institute and the American Lebanese Syrian Associated Charities that supported our work. So thank you so much for the opportunity to be here today, and I look forward to any questions you guys might have. Go ahead.
Thank you. Another really fantastic presentation. You obviously measured VO2 peak as part of the CPET assessments. I was wondering if you measured any or looked at any other CPET variables derived from the breath-by-breath data, like anaerobic threshold or oxygen pulse or ventilator equivalence, and whether they were different between childhood cancer survivors and community match controls, but also in terms of their relationship to cellular senescence?
So great question, and that's the beauty of a maximal test is we have all these other variables that we can investigate. For the purpose of this project, we did not evaluate that, but talking about anaerobic threshold and substrate utilization, there is the suggestion that maybe childhood cancer survivors don't respond the same way. We know that with the muscle phenotype in the general population, we see a reduction in strength followed by a reduction in mass, whereas in childhood cancer survivors, we actually see the opposite. We see a reduction in mass, and they seem to be able to maintain their strength. So looking at those markers from the CPET test, we might be able to understand the connection between the cellular phenotype and the physical phenotype. So stay tuned, hopefully.
Do you have the breath-by-breath data?
We do, we do.
That would be really, really
And then we also have
interesting
the continuous 12 leads, so we have a lot of information we can dig at. So
Thank you very much.
no problem.
Yeah, thank you, Chelsea, for a great talk. I was curious to know if, if any of these cardiotoxic therapies that the patients received had any impact on the thymus gland. Because if there's any damage to the thymus gland, then of course you're relying on peripheral homeostasis of T cells and things like that. So they're going to express more senescent markers like P16 and so forth. And I was wondering if that's maybe something you've thought about.
It is something that we have thought about. We know in the general population, cellular senescence, inflammation, and just chronic conditions in general, we know that those markers are elevated. Given that our survivors do have a high burden of chronic disease, it's likely that their contribution is coming from other impairments in the system, but we didn't control for that in this, but it's definitely something we could look at in the future. Okay,
good. Yeah, and just for one thing, the mechanism, because I'm interested in this also, but if you take interleukin-7, for example, it's one of these muscle-derived cytokines or myokines, and we know that IL-7 has a direct impact on the thymus. I mean, you can inject IL-7, and it will cause immature T cells to be released into the periphery. So I'm wondering if there's a connection there with the release of IL-7 from exercise that's then having an impact on the thymus. It's going to then produce more naive cells that have less of that senescent phenotype, and it could be a mechanistic link there.
I think that's very interesting, and the beauty of working with St. Jude Life and these survivors is that there's a lot that we don't know yet. So there's a lot of room for us to investigate these mechanisms. But on the same side, there is a lot that we don't know yet. We do have a colleague that's collecting a larger panel of inflammation markers in Hodgkin lymphoma survivors. And so we're hoping that information from that in association with exercise capacity, we might be able to tease out a few more of the mechanisms and then come back and revisit this population and dig a little bit deeper. Thank you.
Thank you, that was really interesting. I was interested in some of your patients or participants had high hsCRP levels above 10 milligrams per litre, and I noted that most of them were the female participants. I just wondered whether you delved a little bit deeper into why they had higher levels of inflammation to start with, and in the females, was it purely linked to the fact that they had higher body fat percentage?
So there's two potential reasons. So again, our population of the 185 that we use in this study was across different diagnoses. And so males and females do receive different treatments, so different reproductive organs are exposed to the different radiation fields. And so over the past few decades, childhood cancer treatment is constantly changing. And so it's possible that some of the radiation exposures that the females received didn't affect the male reproductive organs the same way, contributing to some of the endocrine disorders that we see in this population. And so, I think that might be a good thing to note in the discussion is, if we were to stratify this by decades, if we would see the same split difference between males and females. The fact that our females did have a higher level of body fat percent could confound the inflammation marker. But I think the take-home message here is that between males and survivors, inflammation is higher than what we would expect in the general population.
Great,
And that's
thank you.
concerning. Hi.
Really excellent work. You mentioned brown fat and white fat at the beginning, which got me thinking about iris and senescence. So I guess as we kind of figure out and stratify, did you guys stratify, or is there a way to stratify whether it was tan, brown, or white fat? And in the same level, if we're talking about kind of, they may not have been able to do VO2 peak, there was intolerance, I'm assuming, at the highest levels of 70%, but did you get any kind of accounting for physical activity? So maybe just levels of inactivity may, in effect, have changed their irisin levels. So I just was curious about those thoughts. And
unfortunately, we can't get at the different types of adipose tissue from the DEXA scan. We are looking, I am working on a protocol that's using panoramic ultrasound on the quadricep muscle to see if we can get a bit more of the intracellular levels and see if that might influence the contribution of the muscle portion to the VO2 max test. I'm sorry, I forgot the other half of your question.
I was more just, I know there was some at the VO2 peak at 70%, if I think I read that correctly, but I wondered if you accounted for all of their kind of sedentary and non-sedentary time, just to kind of get a sense of whether they were, because that may, in effect, alter some of their...
I think that's a really good point. I think with anything that we're looking at exercise, we need to account for baseline physical activity. In St. Jude Life, we do get self-reported, and I know a lot of people in the audience are going, ooh, self-reported physical activity. We do have that. We're seeing that it's not as reliable, obviously, as an accelerometry. We didn't account for that. We are hoping to repeat this in the larger St. Jude Life study, and discussions have been, okay, we should be having accelerometers, but it was a cross-sectional analysis, so I don't think it would have comforted us too much. But if we were to look at change in an intervention, I think we would definitely have to account for their physical activity outside. Thank you.
Thank you again. Well, thank you very much for this very interesting talk. And now we have Dr. Hemangi Mavadiya, who will speak to us about racial disparity in diet-related perceptions and behaviors among cancer survivors, and she comes from UC Irvine.
Hello, everyone. Good afternoon. Got it. All right. So I'm a third-year PhD student at the University of California, Irvine, and I'm a registered dietitian by profession. My advisor is Dr. Young-Sha Liu, who has tremendous research published in nutrition, epidemiology, and obesity, and she was mentor for this study as well. So my topic is disparity in diet-related perceptions and behaviors among cancer survivors. And what I did was a pooled analysis of different iterations and cycles of this survey called as the Health Information National Trend Survey, which I will now refer to as HINTS. So as some of the previous speakers have spoken about this, that cancer survivors, the numbers of cancer survivors, especially in the United States, have been rising exponentially, and thanks to advancement in treatment, better screening, surveillance procedures, and as of 1st January this year, there were 18 million cancer survivors in the United States alone, and this number is expected to increase to 26 million in the year 2040. Right? So from a public health standpoint, it's very important to research this population and see their, try to improve their quality of life. So the American Institute for Cancer Research, as well as the American Cancer Society, has put forth several lifestyle recommendations, dietary recommendations, physical activity for cancer survivors. And I haven't listed all of them, but just to list a few is to maintain a healthy body weight, have a dietary pattern that's rich in whole grains, plants, vegetables, be physically active. However, something of major concern is that despite having research being shown that adherence to these guidelines are indeed associated with better quality of life and better cancer-related outcomes, it is concerning that cancer survivors are not adhering to these guidelines that are put forth. And so in this research study, which was a survey analysis, I tried to look at perceptions. Perceptions are basically attitudes, knowledge, etc. And I tried to see if there's disparity in terms of race and ethnicity to understand, if there's a difference in the way different racial groups perceive some, diet-related information as well as their behaviors. So a research gap that I'm trying to fill with this study is that disparity in this aspect of diet-related perception has not been studied previously, especially among cancer survivors. And that's what I plan to do by this study. So basically, the hypothesis was trying to fill this research gap, and the main aim was to analyze these diet-related perceptions and behaviors among cancer survivors while controlling for other confoundings like income, education, age, gender, etc. So methods, just wanted to speak a little bit over here. HINTS is a nationally representative survey, meaning if you've heard about NHANES or the US Census, something like that. So they do two-stage probability sampling in order to get a representative sample from the United States population of US non-institutionalized adults, so anybody who's 18 years and older. And what I tried to do is I tried to merge different iterations or cycles of HINTS to get a good enough sample size of cancer survivors, because this survey is just for US adults, but I tried to pull cancer survivors out of that. So I used a total of 8 cycles and merged them by accounting for their sample weights in order to make it generalizable. And I came up with about 3,600 cancer survivors. And so this is just a graphical representation of, how I like the merging process. And, it started from 2003, the HINTS, but I merged HINTS iteration 4 and HINTS iteration 5, which had 4 cycles each. Now, this is a busy slide, but what I just wanted to give you 2 examples of a perception. For example, do you think dietary fiber influences cancer development? This question is a dietary perception-related question, versus if I tell you that what was your fruit and vegetable intake for the past week, that's a behavior-related question. So adherence is mostly a type of behavior. All right, again, this is a busy slide, but I just wanted to summarize that this was just a basic demographic characteristics of my sample of cancer survivors. And we saw that in the white survivors, they were more of older age group versus Hispanics or Black or other minority survivors who were more from the younger age group. Their income level was less than $50,000 US annually. Education level was high school or less for majority of them. So these were just some of the characteristics. All right, to move forward, so just to give you a plan of analysis, what we did was we did chi-square tests, so single factor analysis, and then we did logistic regression. So we adjusted, and we did 2 models. The basic model was just adjusted for age and gender, and then the full model was adjusted for other social factors like income, education, age, gender, and BMI. So this is just chi-square test, and it just shows that there is indeed, disparity or differences between different racial groups when it comes to perceptions about fiber, or perceptions about processed meat, perceptions about junk food, as junk food was defined here as candy, chips, etc. And even there was strong disparity or differences when it came to food label knowledge, which tells us that there's still a need for interventions that target the food label knowledge and education in that regard. Also, so I'll skip to my logistic regression, and I'm just going to summarize the findings. A major finding was that the non-Hispanic Black cancer survivors had significantly higher consumption of sugar-sweetened beverages compared to their white counterparts. And Hispanics had a perception about weight, that weight is something very basic about myself that I cannot change very much. Right? So there were these types of perceptions and there was disparity, distinct disparity, significant disparity when compared to their white counterparts. So yeah, so this is just my basic model and the full model, which was adjusted for different things. So basically, we found that there is disparity when it comes to diet-related perceptions and behaviors among cancer survivors. And this is a cross-sectional analysis, so we cannot derive causal inferences. But this study can really serve as a basis for future studies that can target these groups, these cancer survivors, to tailor an individualized dietary intervention, maybe like a small change, individualized perspective, a pilot study that can help them, develop that dietary changes, behavior changes, and greater adherence to the AICR guidelines. And also, this study can be followed by more focus group and qualitative studies where you actually sit with the cancer survivors, patient advocates, and ask them about the barriers, when it comes to following these guidelines, etc. So some of the strengths of my study was that this was one of the first studies to look at disparities when it comes to diet-related perceptions, specifically among cancer survivors. And it uses a nationally representative sample. So it's easier to generalize this to the larger US population because they oversample the minorities and they look at those aspects in the sampling process. And also, this is the only survey, unlike NHANES is more focused on nutrition, not so much on cancer, but this is focused on cancer, and it has some good perception-related variables that you can analyze for a study like this. One of the limitations was that it is cross-sectional, so you can't derive causal influences, and when it comes to other racial ethnic categories, I had to combine the Asian, Native Americans, Pacific Islanders, and other racial groups as other, because it didn't have enough sample size where I could analyze them individually as an individual racial ethnic group. This past summer, I was selected for the Cancer Epidemiology Education in Special Populations, or the CESP Fellowship, and this study was partly funded by that and was partly funded by the University of California Presidential Endowment Fund. These are my references, and thank you so much. Okay, I'll take a question.
Hi, Bill Klein, National Cancer Institute and the home of HINTS. And I had two suggestions for you, or maybe you can react to them. One is that HINTS has a lot of items in there that could help you drill down a little more and see why these perceptions are different. For example, there are a number of questions about fatalism, the extent to which cancer is controllable, et cetera. And it would be worth taking a look at those as potential moderators of these differences. And then the second thing is, you may or may not know that we recently completed an iteration of HINTS in which we went to 3 cancer registries cooperating with SEER, and those data will be publicly available pretty soon. So that's over 1,000 cancer survivors that you could add to your analysis or potentially replicate your findings. So just wanted to let you know that.
Thank you so much. That's very helpful. Thank you. I'll take from you.
Yeah, so thank you. We're working on a study for cancer disparities, specifically around health promotion. And we just finished AIM-1, where we did some qualitative before we move on to some quantitative data. But a lot of our participants, it was not necessarily a health literacy issue. These were persons of color who were experiencing disparity. It was a lot of access, transportation access. They talked about every few weeks that the SNAP program delivers fresh fruits and vegetables. So I wondered how some of the other issues with kind of our health disparity research may impact the results. And
yeah, thank you for the question.
I would love Dr. Klein to know if we have access, I'm sure from food deserts, but do we have access on the HINTS to that?
Yeah. Yeah, no, thank you so much for your question. And I think it's important to also look at the rural and the urban disparities, and look at, where do we stand when it comes to perceptions over there. And yeah, food access, food deserts is a big issue.
Let me just quickly say the HINTS data are all publicly available. We do have geocodes in the data set. Those are not publicly available, but you can request those. So just find me at the meeting and I can tell you who to talk to about that.
Yes.
Thank you so much. That was really interesting. I apologize if I missed this. Were you looking at any cancer, not specific cancer types?
No, for this particular study, I did not look at specific cancer types. It was just all cancer survivors that I could find.
But do you have those data? I'm just wondering whether it would be interesting to also look at whether there were differences according to cancer types. So maybe when looking at colorectal cancer, they might have been more aware of the impact of dietary fiber intake or red and processed meat.
Yeah, absolutely, because not all cancers are associated with diet. Some cancers are more associated than others, like colon cancer and others. So definitely, that's one area where I can do more sub-analysis when it comes to that.
Right, thank you.
Yeah, thank you so much.
Do you want to join us back for the panel at this point? Are there more questions?
Thank you so much. Thank you, everyone.
Thank you. So we have a whole 6 minutes left for any additional questions. And I just realized that I need to give one more answer better than, first of all, thank you both very, very much. But this question of how we managed to get so many patients to come for the clinical endpoint, for the primary endpoint, this was a clinical follow-up that they had to do after 2 months anyway. So this was like just something I realized I did not respond fully to. So please, do you have any questions for us here?
Yes, there's a couple questions for Dr. Goodenough. The first one is from our online community. Do you think these findings are unique to childhood cancer survivors compared with the general population or individuals living with other chronic disease? For example, HS-CRP is a non-specific marker of inflammation.
Great question. I think the most important thing that's unique in childhood cancer survivors is that they're experiencing these elevated markers decades earlier than the general population. And so they're going to live with these systemic changes or pathologies a lot longer than the general population. And so what we know about these conditions, we've studied from the general aging population, so we don't know what there's going to look like for a child that's coming out of osteosarcoma treatment and coming to the lab at 22, and typically they look like they're performing like they're 40 years old. So I think that's the take home, is that it's decades earlier, and I think we can learn a lot from the general population and in applying it to the childhood cancer survivor population.
One more for you, if I may. Just wondering if you all considered the air quality conditions of the adults that you were studying. As you know, that can be a risk factor for asthma, which could then affect exercise capacity.
I kind of feel like a parrot in saying there's a lot that we don't know. Kiri Ness, who is a member in epidemiology and cancer control and the director of the Human Performance Lab, actually has just completed some geocoding data working, I'm not sure who she partnered with, but we now have the pollution levels for all our survivors based on their zip code. So I know that thought process has come through the greater minds of the accelerated aging, and maybe we can account for that in some of our analysis, or maybe the pollution exposures in our survivors has some sort of additive effect than it does in the general population. But that's a brilliant question, and whoever asked that question, if you email me, I can connect you with the powers that be. Okay, there you go, we will chat. But yeah, that's a great question.
Thank you.
Great. I think everybody had a lot of time to discuss and is ready for some more coffee. So thank you again to this wonderful panel, and looking forward to talking some more as part of this great conference. So thank you.